Okay, dear colleagues, I'm glad to welcome all of you in our conference. Let's start our morning section. My name is Alexander Sebastianov. I will be a chairman in this section today. And first speaker is German Baikalov. Are you ready to present your talk?
Yes, I'm ready.
Okay, colleagues, 15 minutes for each talk, together with questions. Yeah? German, please start.
Okay. Do you see my presentation?
Yes, we see.
Okay. Good afternoon, dear colleagues. Today, I want to represent my topic, the influence of the immobilized subtilisins on the coronary blood in experiment. The circulatory system diseases take a leading position in there worldwide. The largest percentage among them is a coronary heart disease, including angina pectoris, myocardial infarction, and sudden cardiac death. Today, it is actual to search new drugs with the coronary dilating effects, as well as to establish the effects of known and registered drugs on the coronary vessel. In Russia, the drug trombozine is registered and used. The active substance is immobilized subtilisin, which has a direct fibrinolytic property. In the instruction for medical use of the drug trombozine, there is no information about its effect on the coronary blood flow. Since the drug is positioned for the treatment of cardiovascular diseases, we were interested how the immobilized subtilisins has an effect on the coronary blood flow. Test substance. The subtilisins are proteolytic enzymes from the hydrolase class produced by Bacillus subtilis bacteria. The subtilisins are immobilized on polyethylene oxide using access technology. The thrombolytic effect of immobilized subtilisins has already been studied. The aim of this work is to study the influence of immobilized subtilisins on the coronary flow. The Langendorff's method of the isolated mammalian heart is optimal for studying the effect of immobilized subtilisins on coronary blood flow. This experimental model allows to study in detail the physiological and biochemical processes in heart, as well as external influences on it in the absence of systemic neurogenic and humoral factors. In experimental groups, after 20 minutes of perfusion, against in the background of stable cardiac work, the calculated dose of immobilized subtilisin is injected into the glass with the perfusion solution. The glass with the perfusion solution, you can see the number 2. The moment of immobilized subtilisin administration is designated at the point 0 minute, which the coronary flow is considered as the initial flow is taken as 100%. Further perfusion with the study drug is continued for 40 minutes. Coronary flow changes are recorded at the 5, 10, 20, 30, and 40 minutes. In the control group, from 0 to 40 minutes, the perfusion is performed with the Krebs Henselate solution. The study design. The experiments were performed on the 50 male West rats weighing 300 grams. The animals were divided into 5 groups. The control group, the hearts were perfused with the Krebs Henselate solution, and the experimental groups, the hearts were perfused with immobilized subtilisins at the first concentrations. The concentrations you can see in this slide. The first dose you can see on this picture. In the group control, you can see their hearts were perfused with Krebs enzyme solution. We noted an increase in the coronary flow from 5 to 30 minutes, and their increase was maximum 10%. Then there was a smooth decrease to 7% of the baseline. In experimental group 1, you can see there were no significant differences with the group control, but there was a great increase of the coronary flow at the 20 minutes of perfusion. The second group, we noted that from 5 to 40 minutes, we can see the significantly increased in the coronary flow, reaching its maximum at the 20 minutes, and it was 23% compared with the group control. In experimental group number 3, we observed an increase to 4% in the coronary flow by the 5th minute, and then we observed a significant decrease in coronary flow from 10 to 40 minutes of perfusion by 10% relative to the group control. In the group number 4, we observed a gradual decrease in coronary flow from 10 to 40 minutes by 12% relative to the group control. The maximum decrease in the coronary flow was noted at 30 minutes of perfusion. Conclusions. On a model of an isolated red heart, it was shown that with the perfusion of immobilized subtilisins at doses of 170 and 340, an increase in coronary flow is observed, and accordingly the vasodilating effect of immobilized subtilisin. At the dose of immobilized subtilisin 500 and 1000, there was a decrease in the coronary circulation, which indicates an increase in the contractile activity of the coronary vessels. The phenomenon of vasomotor activity of immobilized subtilisin has been established in relation to the coronary vessels. In modern physiology and pharmacology, there is no information about the presence of specific receptors for the immobilized subtilisins in the coronary vessels. The study of this mechanism is planned in the following experiments. Thank you for your attention. I don't hear. Don't hear.
Okay, now, Felix, are you hearing me?
Yes, fine.
Okay, good. Somebody has questions? Yes or no? Okay, I have a short question. I'm not a specialist in cardiology. How do you think your model, experimental model, isolated rat's heart, yeah? Is it adequate model for checking of pharmacological properties of substance? Maybe,
yes. Yes, because this model is unique. Oscar Langendorff made this model very many years ago. Yeah, this model allows to get a direct pharmacological effect, because their hearts has no neurogenic and humoral factors, and this model is this model allows to study this substance,
okay.
subtilisins.
Many things. And other question, I'm not a cardiological doctor, but what is perspective for future application of your results in clinic? How medical doctors can apply your results? Maybe not now, maybe in future.
Okay. When we know the mechanism of this substance, we can use this in the cardiology, in myocardial infarction, endothelial dysfunction, maybe their thrombotic occlusion, maybe in the angina pectoris.
Okay, thank you. Thank you for your presentation.
Thank you.
Next speaker in our program is Kurochkina Yulia. Is she present? Yeah, Yulia, are you ready to present? Yeah.
Yes, of course.
Yeah, please start your presentation.
Good morning, dear colleague. I'm glad to tell you about rare autoinflammatory diseases. Do you see my presentation?
Yes.
Mm-hmm. It is very important in clinical practice because it's very hard to make this diagnosis, because it's necessary to have sequencing technique, and it's very expensive nowadays. It's not easy for every patient. Autoinflammatory diseases are a group of diseases that include familial and Mediterranean fever, TNF receptor associated periodic syndrome, Macula syndrome, and others. The cause of these diseases is a mutation in one gene. It is monogenic diseases. A mutation in the gene leads to inflammasome and leads excessive activation of caspase-1, formation of inflammasome, and hyperproduction of interleukin-1. Elevated concentrations of interleukin-1 cause the main manifestations. Clinical manifestations include urticaria, rash, fever, arthralgia, arthritis, sensorineural hearing loss, and kidney disease such as amyloidosis and death. In clinical, for treatment, we use non-steroid anti-inflammatory drugs, corticosteroids, antihistamine drugs, but it's not effective nowadays. The main effect can give us monoclonal antibody against interleukin-1. There are two drugs in Europe. It is anakinra and canakinumab, and it's only canakinumab in Russian. I would like to present a case history. We use sequencing technique for revealed mutation in gene NLRP3 for Macula syndrome. And we investigated also the gene TNF-RS4. It is connect with TNF receptor mutation. It is a case history of 31-year-old woman who was admitted in our clinic, and she complained on arthritis, urticaria rash, and fever. Their onset of disease was at the age of 7. After that, it was appearance of arthralgia, arthritis, relapses of conjunctivitis. She was examined in allergologists numerously, but the main diagnosis was recurrence chronic autouria, and the treatment was antihistamines drugs, and it was not effective. She undergone genetic testing because her children have Muckle syndrome. It was proved by sequencing technique, and they receive canakinumab. You can see autouria rash. It always present on her face, body, her arms. It is family tree of patient with CAPS syndrome. This is our patient. This is her children. They have proved Muckle syndrome, as I said. This is her own sister. I will say about her some minutes ago. And this is her mother. Unfortunately, she died some months ago. She had fever, autouria rash, and kidney disease. And we suppose that the main cause of death were amyloidosis. We examined this patient in our clinics. We revealed recurrence of autouria rash, recurrence of conjunctivitis, the elevation of systemic inflammation markers such as ESR and C-reactive protein. Her urine analysis was okay, and she has normal renal function. She has not only pathogenic variant of NLRP3 associated with cryopyrin syndrome, but she also has TNF-ISFR mutation. It is connected with TNF receptor mutation, but based on clinical features, we understand that it's cryopyrin-associated syndrome, Muckle-Wells. This is history of her sister. She also has autouria rash, but she had the onset of arthritis during the pregnancy. She was examined in rheumatology, but rheumatoid factors, CCP, anti-nuclear antibody were negative, but she has a diagnosis rheumatoid arthritis, and she receives methotrexate in 15 milligrams per week with good clinical effect. Nowadays, we clearly understand that it's not a real rheumatoid arthritis. We suppose that it is symptom of her main disease, Muckle syndrome. She also undergone genetic testing, and she also has a pathogenic variant of NLRP3. But nowadays, she has only recurrence of autouria rash. She has not got any arthritis nowadays. The levels of ECR and C-reactive protein normal, and she has not got any kidney disease nowadays. It is a genetic cut, as I understand. The sequencing were made by our colleagues from sector of genomic mechanisms of ontogenesis. And as I understand, we can see stop gain in such location. It is about NLRP3 and TNF-SVR, but the main diagnosis is, of course, cryopyrin associated syndrome Muckle-Wells. About treatment, it's very hard nowadays to receive canakinumab for elderly patients because it's easy for children, not very easy, of course, but all children who need in such kind of treatment, they receive these treatments, but it's very hard for their parents, and nowadays they could receive only non-steroid anti-inflammatory drugs, corticosteroids, but his mother received canakinumab after her children because the dosage depends on weight, and nowadays he could such possibility, but we work with this team. We try to have an agreement with company and try to do sequencing technique in our sector to prove such kind of diseases. Thank you for your attention.
Thank you for your talk. Somebody has any questions? Unfortunately, no. Thank you for your presentation, Yulia. Okay, next speaker is Sergey Staroletov. Is he present? No, yeah, we have not. Oh, I see Sergey. Sergey, are you here? Sergey.
Yeah, I'm here.
Oh, okay. Thank you. Are you ready?
Yeah, I can share my screen.
Yeah, sure. Please start.
So I have some technical issues.
Yeah, sure. Not a problem.
Okay, and
yes, we see.
Yeah.
Yeah, I see your presentation.
Okay, I will start. My name is Sergey Starletov, and I'm from Altai State Technical University. And also I work for Institution of Automation in Novosibirsk. And I'm going to present a hierarchical temporal memory model in the cells of Hawkins. And in this talk, we are going to present some overview of related literature. We present HCM concepts, and we present our HCM physical model and its visualization software. And then we move to temporal events organization in the case of music. And finally, we will present HCM logical models and our concept of smart audio studio. And this paper is related to ideas of Jeff Hawkins. Jeff Hawkins is a creator of Palm computing, and now he moved to science related to brains. And now he's one of independent brain researcher, and he produces some software for analyze anomalies in network data. And his work is about brain neocortex, and we can see that in our brains we have a special superficial cut of the brain. Of the top of the brain, we see neocortex, and he stated that it is responsible for producing invariance and hypothesis about the data. So neocortex is a device, is a layer which provides us some intelligence. And he published his famous book on intelligence, and it has also translated to Russian, so we can read it in Russian. And there are a lot of generalization of current state of the brain research, and he presents our concept of hierarchical temporal memory, and he states that the brain constantly works as a memory predictor, and this model is nothing related to modern artificial neural networks, so it's a brain-inspired model. But no algorithms were given explicitly in the book, so then he published a white paper on HCM concepts, and it's also been translated to Russian, and there is some overview of cortical algorithms here, and he presents algorithms of work of spatial and temporal pullers here. And then this year, he published a new book, 1,000 Brains, and it encourages me to write this paper. So he states in his new book that there is no single model of an object in the brain. So the knowledge is distributed into thousands of models in the brain, and we have a lot of prediction systems. They work as a worker in organization, and the recognition process is a process of distributed voting here. So Hawkins team, they produced some software. Some of them are open source, and you can see that Nument and Nupic, this set of C++ classes and Python files. so it is usable. And he also produced paid software like Grok, and we can use also Numenta HCM Studio for academic licenses to just to visualize some anomalies in data. But now we can see that there is some competitor from modern AE side. It is LSTM concept, and it produces some ideas, but they are implemented as recursive neural network. So this concept has forget gates for short and for long memory. So we can see that nowadays some ideas are also implemented in modern AE, but in this work we are going to speak about brain-inspired models, because LSTM, it is an artificial model. And we can say that HCM principles like presented on the slide, and there are some principles like temporal organization, blocks, auto-associative nature, and the ability to step in information, invariance generation, and significance. Invariance generation is the main problem for studying, because even Hawkins said that it is a mystery how the brain forms our invariant representations. If we describe a physical SGM model, we have 6-layered cortex, and the top layer is responsible for sensor processing, and then we have connections between neurons to several neurobiologists, and they have some longitudinal synapses connections through all the layers. And they are connected to some sensors that are able to produce sight, smelling, hearing, and feeling. And then synapses can be connected with proximal connections as well as distal connections, and cells formed a column which consisting of an average of 10,000 cells. And you can see some representations that were depicted when we studied it model with my student Dennis. And then the main innovation of hierarchical temporal memory is concept of spatial and temporal poolers that are responsible for grouping or generalizing the data. And the spatial pooler allows reducing the data from the sensors to a sports representation, while the temporal pooler builds internal representation based on data received from spatial pooler and forms invariance. And we produced some software that works as an OpenGL software, and it can process some bit vector. And we are implementing some physical HCM algorithms to work like spatial and temporal poolers, so one can visualize the activity of neurons in a cell by a given bitwise vector. But if we work with some physical models, it's hard to produce some algorithms for form invariance and detect and produce some real models responsible for real events processing. So we then moved to some kind of logical models of HCM memory. And music is a good example of signals over time that the brain can tune to. And we work with MIDI files, and each MIDI file represents a sequence of notes. And it's important that intervals in music are very significant, and real musicians must respect both the tonality of the melody and its timing. And we can see that my student is a musician, Nikolai Chernov. He played the same melody like a hand played, and then it constructed the real melody from notes, and we can see some differences in two plays. For example, in a manual play, software should insert some intervals to respect the correct events. Like you can see, we have a timing, and we should respect this software should respect this timing. So, like our brain works, he can change some errors in events, and he can tune to some timing, and the brain can correct this timing. And if we degrade the quality of two fragments, like a real fragment and a played fragment, and we can see that if we degrade the quality, these segments will become identical. So, which makes us think that invariant form is just a value representation of a sequence of signals. So it determines the ear of music for a particular human by the degree of accuracy to able to recognize such a sequence. And if the brain is more trained, it will notice a lot of deviations, since it will make a lot of unreasonable predictions. And towards a smart audio studio in Intel school, we as a group of students 10 years ago implemented software that respect Hawkins' ideas, and you can see some images of it. And it was not completed, but we worked out the basic algorithms for processing MIDI sequences and forming a melody trace in the form of hierarchical temporal memory. So we are able to produce graphs for a given melody. And you can see one graph and another graph for the same melody. It can become identical if we play with some parameters. So we produce some ideas, and I depict some ideas in this slide. So we have a moving window for short memory of recently played notes. We have a moving window of current hypotheses, and we constantly try to reduce a set of notes and current hypothesis to an existing hypothesis. And we are able to arrange the input events, and we use Boost Graph library, and we use design patterns, so we can substitute some strategies of calculation of delta between musical events. So now I started to improve this software, and you see my student, he is a musician, and he now improving, and we will some kind of debug HM memory, and we can produce some internal representations of HM memory at each step of play. So we can debug the parameters and debug the algorithms and produce more correct representation of digital graph from melodies. It can be used for melody comparing, and it's kind of research, because subject of research, because I can conclude that we reviewed and we state that we can use bio-inspired algorithms, and we should also respect new ideas from Hawkins. But my some intention that we can use these concepts to study, to search for anomalies in incoming data in event-oriented cyber physical systems. So, like in data from CAN basis of automobile devices. So it's some kind of new direction of my research. Thank you for your attention, and I'm happy to hear some questions and respond them.
Ведущий, вас не слышно. Включите микрофон.
Okay, now it is working.
Yeah.
May I?
Okay. Yeah, thank you.
Sergey,
Yeah.
this is not a question. I would like to say thank you to you for very interesting research. I tried to use music for medical biopsy interpretation. And I have one recommendation. I say that for you in Russian. Посмотрите работы Владимира Александровича Лефевра по музыкальным интервалам на основе работы мозга. Это очень интересные работы. Thank you.
Okay, thank you for the comment, and I will send your comment to my student because he is a musician, and along with him we can understand, I think, these ideas and implement it in the form of software.
Okay, any other questions? Okay, Sergey, I have questions. Very interesting model, but it's a theoretical model. Do you know any experimental studies of real human or animal brain which support this model, or it is just a theoretical model without experimental support?
About my model, I can say that I have like two models, and I'm going to combine them. Like I have a physical model and I have a logical model, and sometimes I think we will combine it, but it is not just evaluated correctly. I think it is now in a research state, but if we browse papers by Hawkins, he has a lot of papers, and he has some papers of study of the significance of his models with a comparison to some competitions. So he published a lot of papers on the evolution of such models.
It's evolution of models, but do you know at least one experimental study in real brain?
I can say that he has a Grok program. It is, as I depicted here, it is related to processing of TCP/IP
Okay.
data. So he is able to produce some kind of anomaly detection for network traffic.
Yeah, it's not real physiology. Yeah, it's just computer model. Okay, thank you.
Yeah.
Any other questions? Okay, thank you for your presentation.
Thank you for the ability.
Okay, our next speaker, just a moment. Filimonov Vyacheslav, please, could you present your talk?
Yes.
Okay, very good, but speak English.
Yes, of course.
Sure.
Do you see my screen?
Yes, we see.
Thank you. In general, our article is about abilities and how to develop it. But I analyze the program of our conference and think it will be better to tell about interesting experiment and very brief about our approach. The approach is named 4C, and there are concepts: collaborative, collective. And if you are only one person, you may simulate collective work, for example, using six thinking threads of Edward de Bono. And there are three another concepts, and I would like to recommend you Edward Tufte, cognitive graphics, Vladimir Alexandrovich Lefebvre, configurable reflexive analysis, and Corgan-Gent work of team by Alexander Rykov. So there the first slide about our experiment. It is an interesting phenomenon, uncanny valley. It was developed by Masahiro in 1970 in Japan, Kyoto University, and in 2012 in English. There were many investigations that detailed the results, but the main result is it was hypothesis that the more a robot is like a human being, the more sympathy a man can feel to it. But it will be some distortion named uncanny valley. And it was interesting to understand why the distortion is acquainted. And we think that there is some hysteresis effect. You may see it in experiment on the stability of perception, of visual perception. You may see the text in the theory of catastrophe in the book of Stewart and young Stewart, yes. And we think that we may use this theory to explain this effect. And here, our example, not picture, but some words. And when you read it from left to right, you may see that the word cop read one in English and it means a policeman. And when you read the second sequence, this is sword, this is fresh in Russian. And secondly, we think that a human being may be dangerous too. And there may be some another scale, and we try to use it. This is a model we think at first as witness assembly. This is demonstrators association with the anxiety, arousal, and performance. This is first results, but I think this is much more understandable. This is a book of Ian Stewart, and so this is it is only in French, not in English. So I tried with a Russian explanation. This is a Whitney assembly. You see, if you move from to left bypass of bifurcation points and right bypass bifurcation points, you get another results. We used such a checkboard matrix where two scales scales as human likeness and scales of threat safety. It was our scale. And the third dimension was the sympathy, was objects like such as this slide. This is the matrix of our experiment. Here are 25 pictures. They're in the order of our scales from human likeness, from left to right, and from safety to threat, from up to down. And the experiment implementation was when all these 25 objects, one for one, were shown on a large screen. And participants had some special blanks, fonts, and they put their marks on it. And different groups gets different sequences, left bypass of bifurcation point and right bypass. And there are more than 120 people, from young as 14 or 35 students, Omsk and Vladivostok. And there is one of typical result of groups, and we see their estimates are different, and the bifurcation points is a very is a point that makes different ways get different estimates. So this is we have the hysteresis in this research, so it's enough for us. No, thank you for your attention, and we candid uncanny valley is a two-dimensional projection of a 3D assembly. Well, in the paper we have a description of our another experiment, but now it is I have no time, and I see no use to tell about it. Thank you for your attention. I am ready to hear your question, and speak slowly, please. I have some problems with my hair.
Thank you for your presentation. Somebody has questions? Okay, thank you again. Thank you for your presentation. And the last presentation before break, Evgenia Privatnova. Evgenia, are you ready?
Yes, hello.
Hello.
Okay, my topic is difference. Do you see the presentation?
Yes, sure.
Everything okay. Difference in associations of polymorphic variants of the serotonin transporter gene with resting state AG in younger and older adults. Mental aging is a heterogeneous process, and resting state brain activity is suggested to reflect tonic brain activation that supports basic physiological properties of brain functions and age-associated changes. Genes involved in the regulation of synaptic function are candidate genes to be associated with characteristic of baseline EEG activity. Serotonin projections are widespread in the cerebral cortex, and serotonergic network is highly plastic, showing changes in its anatomical organization over the lifespan. The activity of serotonergic system which plays an important role in plastic rearrangements of brain structure and activities throughout life, can be considered as one of the factors in the heterogeneity of brain aging. And one of the key factors regulating the activity of the system is the SLC6A4 gene, which encodes the serotonin transporter protein, which ensures the reuptake of the neurotransmitter from the synaptic cleft. Two functional polymorphisms of the 5-HTT gene have been identified. One of the most studied polymorphism is in the promoter region, 5-HTTLPR, with short S and long L alleles. High expression is associated with LA, LL, and low expression with S allele and LG variant. The second polymorphism is caused by a change in the number of tandem repeats of INTR in the second intron of the serotonin transporter gene. It's called STIN2 VINTER with 9, 10, or 12 repeat alleles. The STIN2 VINTER polymorphism acts as an allele-dependent enhancer of serotonin transporter gene expression. The effects of STIN2 VNTR polymorphism are less well understood compared to 5-HTTLPR, especially with regard to its association with changes in brain activity. And the combined effect of 5-HTTLPR and STIN2 VNTR on the characteristic of the background brain activity hasn't yet been studied, especially taking into account age differences. Therefore, the purpose of the present study was to identify associations between combined effects of two polymorphisms of serotonin transporter gene. They are 5-HTTLPR and STIN2 VNTR, and the resting state AG, and to investigate whether these activity patterns are moderated by aging. Since baseline resting state brain activity predisposes subsequent cognitive performance, we proposed that genotype-dependent differences in resting state EEG might mediate properties of attention. The sample included a young age group, 217 adults from 19 to 35 years old, and old age group, it's 107 adults over 55 years. Participants were students or full-time employed subjects. They had no mental disorders, traumatic or other brain lesions, or major medical diseases, including cancer, cardiac illness, or diabetes. All subjects gave informed consent to participate in our study. Participants visited the laboratory twice, once for EEG recording and another one for psychometric assessment, which included the attention network test and modified oddball task. The EEG data were registered using 16 HH Chlor electrodes under eyes closed and eyes open conditions. The EEG spectral density was calculated using fast Fourier transform and 8 individual frequency ranges of delta, theta, alpha 1, 2, 3, beta 1, 2, and gamma rhythms were determined using the individual peak power of the alpha rhythm. For genotyping, we extracted DNA from buccal cells, and polymerase chain reaction was used to detect S and L alleles of 5-HTTPR and 9, 10, and 12 alleles of STIN2 VNTR in the DNA samples. Next, we detected LI and LG polymorphism. We grouped LG and S alleles given their functional equivalence. Based on our previous behavior data and based on the idea that expression of 12 allele is lower than 10 allele, 3 genotypes combinations were created. The first one is a combination with high expression level, no low expressing genotype at ACE of the loci. The second group was intermediate. A low expressing genotype was only in one at the loci. And the third was a low expression group, and low expressing genotypes was at both loci. For statistical analysis, we used ANOVA for calculating differences in ice-close and ice-open EEG power data, and to assess if resting state Bray electrical activity mediates the effect of serotonin transporter gene expression level on indices of attention. We tested simple mediation models created using process with SPSS syntax. Okay, for all analysis, genotype-associated difference in EEG spectral power was found only in older adults and was absent in the young group. These findings are well consistent with the suggestion that genetic differences have an increasing impact on brain functioning when resources decline from higher to lower levels during the process of aging. For the eyes closed condition in older group, carriers of combination of genotypes with high expression had lower power compared to low expression and intermediate expression in alpha-2 and alpha-3 rhythms. For the background EEG activity with eyes open, again, carriers of combination of genotypes with high expression had lower power estimates of alpha-2, 3, and beta-1 rhythms localized in the central, temporal, or parietal occipital regions. In the right figure, we can see the data for central temporal region, and we can see from the figure that high expression group in older age group had significantly lower power compared to low expression group, intermediate expression group in alpha-2 rhythm, and the same results in alpha-3 and beta-1 rhythm. At the second step of our analysis, we chose variables for mediation models. First, we performed the correlation analysis between the EEG estimates and indicators of attention only in older group. Significant correlations were found only between EEG power under eyes open and orienting reaction. Therefore, we selected them as our cognitive indicator. The genotype-dependent difference in EEG power in older group was found for alpha-2, 3, and beta-1 rhythms localized in central, temporal, and parietal occipital areas. Therefore, we took these EEG estimates as mediators. And since high expression differed from the other genotypes, high expression group was used as reference group within dynamic coding system for multi-categorical independent variable. Our results are presented in the table. Here we can see the mediator in alpha-2, alpha-3, and beta-adene rhythms in different areas. We can see here the dynamic coding system of independent incident variable and relative indirect effect. AB coefficient means multiplying a regression coefficient of path from incident variable to mediator with the regression coefficient B of path from mediator to outcome variable. For statistical inference about indirect effect, percentile bootstrap confidence interval with 10,000 bootstrapping samples was used. And significant effects are in bold. We revealed a significant indirect effect linking serotonin transporter gene expression level to orienting reaction through spectral power in alpha 2, 3, and beta 1 rhythms are localized in central temporal, parietal, occipital areas. In all cases, serotonin transporter gene expression level was not directly associated with orienting reaction. As tested mediation models were similar, we presented only one model for alpha 2 rhythm in the next slide. Here we can see negative indirect effect. That is high expression group relative to intermediate expression and relative to low expression was associated with lower alpha 2 power, which was translated into more pronounced oriented reaction. Alpha power increase is proposed as the mechanisms through which task-relevant information is suppressed. And accumulating evidence suggests that alpha power has age-related reduction. Inability to modulate alpha power, therefore, may be a basis from deficits of ignoring irrelevant information and destructibility that are common findings in the older age group. Based on those results on our analysis and from the literature review, we can suggest that older group of high expression level, which demonstrated weaker alpha 2, 3 power in the posterior cortex, might have limited ability to ignore destructive information and therefore has higher responsiveness to novel task-relevant features of the stimuli. And supporting this suggestion, we found that AG properties mediates the effects of combined 5-HTTL-PR and STIN2-VNTR genotypes on orienting reaction, so that high expression genotype was associated with low estimate of alpha 2, 3, and beta 1 AG power, which in turn were translated into more pronounced oriented reaction, meaning more responsiveness to novel and task-relevant features of stimuli. Thank you for attention.
Thank you. Thank you for your presentation. Somebody has questions? Okay, I have a question. Distribution probability of different alleles in your groups of subjects were same or might be different?
We tested the distribution according to Heide-Weinberg, and
Okay.
all distribution were similar to Heide-Weinberg.
Yeah, no
In
differences.
the
Okay.
joint group and in older and in younger.
Okay, thank you. And also, how you estimate which statistical method you apply for estimation of mediative effect?
Can you repeat the question about?
You told me that EEG in temporal area mediate effect between genotype and orienting reaction, yeah?
Mm-hmm. Yeah.
But how you estimate this mediation?
We built mediation models in process application to a specific statistic. So our statistical model we can find here. We have got independent variable. We use dummy coding system. So we compared a low expression level with high expression level and intermediate expression level with high expression level. Therefore, we have two independent variables instead of one multi-categorical variable. And our mediator were 1, 2, 3, 4, 5, 6 mediators and 6 models. 6 mediator models.
Thank you. The last question. Initially, in Lesch and his course works, this polymorphism was presented as a candidate for depression and other affective disorders. Yeah, but in your studies, in our studies, we see that very different relationships between this polymorphism and behavior, different relationships between this polymorphism and EEG in different subgroups. Yeah, you see that it depends on age. I see that it depends on nationality and region. Yeah. And climatic region. And in your view, how do you think, can we apply this polymorphism as a marker of disorder in screening of mental disorders? Yes or not? Is it possible to apply this polymorphism as a marker of, for example, depressive disorder?
It may be one of the markers, but not the main one, I think, because we can find a very complex relationship between genotype and the outcome.
Yeah, and effect is very changeable. Yeah, it's strongly depends.
As for this study, Alexander, as for our study, we found effect of this polymorphism only in old age group. And it seems to me very logical because when the person became older, all genes have a greater influence because cognitive reserve, which can play a big role to affect effectivity of performance decline, and genotype can greatly influence.
Okay. But Lesch has found effects not only in adult group. Yeah, he found not only in old people group. He reported about this effect in young group. Yeah, but for example, there are strong contradictions between American studies and Japanese studies. Yeah, in our also a strong contradiction between studies in Europioids and moncholoids. Yeah, it means not only age influence to such relation, and it's very difficult to apply this polymorphism for diagnostic because strong interactions between this polymorphism and many other factors. Okay, thank you very much for your presentation. It was the last presentation before break. Yeah, now we will have break till 12:30. Yeah, 12:30, and after break it will be presented very interesting talks from other countries, from Taiwan, from Turkey, from Germany, from USA, and I recommend to all of you to participate in prolongation of our section. Thank you for your participations, for your attention, for your talks. Now we have break for 20 minutes. Okay? Break. Dear colleagues, are you hearing me? Microphone is working?
Yes, fine.
Okay, fine. Next speaker, Ludmila Yansholi. Ludmila, are you ready?
Yes, I'm here.
Sure. Please start.
My presentation is okay?
Yeah, sure.
Hello, everyone. My name is Ludmila Yansholi, and I am a scientist from the International Tomography Center here in Novosibirsk. And thank you for the opportunity to tell you today about metabolomic profiling of biological fluids of patients with lymphedema. Despite the high diagnostic level of modern medicine and modern medical technologies and instruments, there are still some diseases that are not easily diagnosed and prognosed. One of them is lymphedema, or so-called elephantiasis. This is the accumulation of protein-rich fluid in extracellular space under skin and subcutaneous tissue. Lymphedema is an external or internal manifestation of lymphatic system insufficiency and deranged lymph transport, causing the chronic inflammation in human body, adipogenesis, fibrosis, and as a result, the increasing of volume and disfiguration of upper limbs, arms, and lower limbs in men and women, in women especially. There are two causes of lymphedema. One of them, this is a congenital lymphatic dysplasia, also called primary lymphedema. This is very rare case. It's just one patient per 100,000. And today we will talk about secondary lymphedema. This is acquired obliteration, such as after radical operative dissections. Here on this slide, I should show you not very pleasant, but very important pictures that show stages of lymphedema. The first stage, where we can see damaged lymph vessels, but no clinical manifestation, with the early and not very high accumulation of protein-rich fluid when the edema is pitting. The second picture, this is the second stage, which divides to two parts. This is the early stage, when the protein-rich fluid in interstitial accumulates, but edema is still pitting. And the later stage, when the lymph doesn't pit because of excessive subcutaneous fat and fibrosis. And my samples for today, this is the stage 2 of lymphedema in women. And the last stage, very important, this is the stage 3. This is very dangerous stage with chronic inflammation in human body, with persistent accumulation of metabolism products and further deposition of fat and fibrosis and warty overgrowth development. I should say that the most important reason of lymphedema is breast cancer. And here I show you some statistics. So you can see that about 20% of lymphedema after breast surgery is the axillary lymph node dissection, about 6% is the sentinel lymph node biopsy, and any type of regional nodal radiation may lead to lymphedema. This is an increased risk of lymphedema in future. This is foreign statistics, and what we can say about Russia and Novosibirsk particularly. Our colleagues in the clinics of the Institute of Clinical and Experimental Lymphology have patients with lymphedema. This is mostly women, which have breast cancer surgery and radiation after in their anamnesis. So this all speaks in favor of relevance of this research for the world science and for the Novosibirsk particularly. This is a very important phrase that was published by the Executive Committee of the International Society of Lymphology last year, and it sounds like research in molecular lymphology, including lymphatic system, genomics, proteomics, metabolomics, and systemomics should be greatly expanded. It means that there is some information about genetics of this disease. There are papers about proteomic of the interstitial fluid in lymphedema, but there is still lack of information about metabolites and metabolomic changes during lymphedema development. And it means that the systematic information combining genetic information, proteomics, and metabolomics may be very important for understanding the etiology of this disease. Here, I show the pictures from our colleagues from the Laboratory of Operative Lymphology and Lymphatic Detoxification here in Novosibirsk. And on the left-hand side, this is an ultrasound examination of lymphedema in arm after breast cancer treatment. And here you can see the non-affected limb. And on the left-hand side, this is a affected limb, where here with black, we can see the accumulation of free fluid in the subcutaneous tissue. Also here, we can see in dark the cavities which contain free fluid that accumulates in edema, and this is called lymphedema. Sometimes it is possible to extract this free fluid from patients, not from every patient, but sometimes it is possible. And how does it look like? So you need to be very precise to get into this cavity to extract interstitial fluid without mixing with blood, because our research is aimed to compare human serum with human interstitial fluid to make it possible to find some unique or complex biomarkers for prognosis and diagnosis of lymphedema with metabolomic approach. Some words about metabolomics and metabolites. Metabolomics, this is a young branch of post-genome sciences. So metabolomics is about small molecules under 1,000 Daltons. This is amino acids, antioxidants, nucleotides and nucleosides, organic acids, lipids, sugars, and osmolytes. There are different approaches in metabolomics. So the not very interesting one, this is a qualitative metabolomics, when you can say that sample A contains alanine, for example. The more interesting is semi-quantitative approach, when you can say that content of alanine in sample A is twice as large as in sample B. And the most interesting one, this is a quantitative approach, when you can define the exact concentration of alanine per gram of tissue or per milliliter of biological fluid. So you can get the metabolomic profile of, for example, normal tissue or fluid, compare it with pathological metabolomic profile of tissue or fluid, find some metabolites that are different in these two cases, and then looking on the map of metabolic pathways, you can find these metabolites, for example, here, here, or here, and say that changes in concentrations of the metabolite here may lead to changes in concentration of metabolite here that ends with the changes of the concentration of end product. Another way and another approach, metabolomic approach, is the comparing of pathological tissue or fluid with pathological tissue or fluid treated with drug. So in this case, you can say which metabolic cycles and pathways were disturbed, which pathways are affected by the drug, and is it possible to back the metabolomic profile of pathological tissue after drug treatment to normal one. In our case with lymphedema, we will compare pathological human serum with pathological human interstitial fluid to make an attempt to find some kind of biomarkers for prognosis and diagnosis of this disease. We have these samples from one patient, make the metabolomic extraction, have the extract, and then we have two ways how we can analyze this. NMR method is the quantitative one, very straightforward, when you can calculate the concentrations of most abundant metabolites in fluid or tissue. For now, it is made in this way is made, so we measure the NMR spectrum of serum and interstitial fluid, and the second way is HPLC-MS analysis that is not made to now, but in this way you can analyze hundreds of metabolites in semi-quantitative mode. And this some preliminary results that we have. So this is the representative NMR, proton NMR spectrum. All lines are annotated with our homemade database, so we can say which line corresponds to which metabolite. And this is the results in concentrations. So we measured about 40 metabolites in both in serum and interstitial fluid of one patient. And at first, I should say that in this patient, we found a very high level of glucose. And in green, I marked the acids, organic acids, which concentration in interstitial fluid is higher than in serum. So, and in yellow, the acids that which concentration in serum is higher than interstitial fluid. And for example, these metabolites may serve as potential biomarkers for prognose the lymphedema in patients. But to be confident, we need much more statistics to make the very good results. And our colleagues from the Laboratory of Operative Lymphology and Lymphatic Detoxification, they have some more samples of serum and interstitial fluid, and this work will be continued soon. I think till the end of the year, it will be finished. Thank you for your attention.
Okay, thank you for your presentation. Any questions to Ludmila? Is, who wants to ask questions? Okay, thank you again. Thank you for your presentation.
Thank you.
Next speaker, Volkov Mikhail. Is he present? Okay. He is absent. Okay. Евсеев Петр. Петr, are you here? Петр, I don't hear you. No sound. Please switch off. Yeah, наушники уберите.
Can you hear me now?
Oh, yeah, sure. It's okay.
It's great. Hello, everybody.
Hello.
Please, it takes a second. Do you see my screen?
Yes.
Great. So, hello, everybody. Thank you for the invitation for participation in this conference. Thank you. And thanks to our organizing committee. Thanks to every participants. And today I'd like to tell you about our studies, to report some preliminary results. The subject of our research is the presence of ribosomal RNA-like regions in gene bank viral sequences. We think it's an interesting subject for research because we know some reports about translational apparatus found in viruses, but we know only about ribosomal proteins and some very occasional reports about distant related ribosomal RNA sequences in viruses. Actually, viruses do not have to possess ribosomal RNA sequences, they do not use translational apparatus like bacteria or other cellular organisms do. However, one sunny day, when we analyzed phage genomes, we work with phages, we work with phage therapy. We were wondering that we found in a decay phage DP10 annotated ribosomal RNA sequences. It was very peculiar, and we wrote and sent email to our Poland colleagues. Our Polish colleagues confirmed their results. They answered us that they checked twice, and they really did find such sequences in a bacteriophage genome. After that, we checked the GenBank viral database and found another virus, a bacteriophage, Pseudomonas phage PN05, where an 16S rRNA sequence was also annotated. And after that, actually, we decided to check all the data available in GenBank in different GenBank databases. The first step was the selection of cellular organisms used for the search. We used 80 sequences. We extracted them from annotations, and we found them with known pipelines. Those sequences were related with different organisms from very different taxa, including eukaryotic organisms, bacterial organisms, mitochondria, archaea, and nucleomorphs. You can see the phylogenetic tree made with sequences of ribosomal RNAs. The next slide will show how we searched for sequences which can give us more information about their origin. We were wondering and amused that the GenBank databases contain a lot of sequences related to ribosomal RNA. Actually, Blast Search gave about 3,000 results belonging to 413 organisms. This yellow, this green circle is 413 organisms we found in the biggest database NRNT. We analyzed these 413 entries and divided the sequences into two parts. One of them contains sequences with higher GC content, more than one-third, about one-third, but more entries contained sequences with a low GC content, basically including A and T nucleotides. And we decided to work only with GC content rich sequences. We analyzed those 56 sequences and sorted out 33 sequences because they are too short. So we used a threshold of 100 nucleotides and used for the following analysis sequences of 100 nucleotides and longer. Finally, we analyzed those 23 sequences and found out that some of them are misannotated. We found very suspicious similarity with mycoplasma sequences and ribosomal RNA sequences. For example, we found 8 viroids, and those 8 viroids were exactly the same as mitochondrial sequences from organisms they were extracted. So we thought that was just a wrong assembly. In other cases, they were just misannotations. As a result, we found 14 entries from the biggest NCBI databases. database, and also we found several entries from other databases. On this slide, you see a table which contains sequences we found in viral genomes with a length of 100 nucleotides and more, and similar to ribosomal RNA sequences for small subunit RNA and large subunit RNA. You see that they include both RNA viruses, DNA viruses, and they were extracted from identified organisms as well as from environmental samples. Here are the examples of these sequences. First, take a look at the sequence on the top, the upper sequence. You see that rich AT content is not very nice for analyzing. We get some similar formula, similar regions, but it's not really possible to say about anything about their origin. So we don't use them for analysis. However, some of them are very interesting because they show a great similarity with RNA sequences in cellular organisms. Please also take a look at Blutan virus isolate. That's an interesting example of how mycoplasma, which is ubiquitous, which is everywhere, can cannot it can help us. It's a pollution for our assemblies. However, two viruses here, Marxella FH MCAT16 and grapevine viruses, are very appropriate for further analysis. After that, we used more reliable sequences and made blast search against different databases containing cellular organisms. What we found on this table, you see the virus and you see the organism where it was found, or the closest organism possible. The conclusion from this table, from the search, is that we find mostly the sequences similar to the sequences in the host. If we find an RNA sequence in a virus, it's practically always a sequence similar to an RNA sequence in a viral host. Sometimes it's not a viral host. Sometimes it's another organisms of the same ecological niche. The most interesting example is a stealth virus. Stealth virus is a herpes virus. It was found in different organisms, including human, monkey, and other. And it was shown that these organisms, it was shown about 10 and 20 years below in several studies, that this organism incorporates bacterial sequences. Unfortunately, the authors of those studies did not analyze for RNA sequences. When we analyze viral sequences, regular pipelines don't analyze for RNA. But we did it, and together with other bacterial sequences, we find an RNA sequence in this virus. You see here the comparison of viral sequence upper and a bacterial sequence below. Colored lines are disagreements between two sequences. We see that there are just few of these lines, similarity is high. What we did next, we made phylogenetic tree. We used phylogenetic methods to find out possible related organisms to stealth virus RNA. As well as in previous study, it was an Ochrobacter, Ochrobacterium sequence belonging to this organism, but not exactly the same. It looks that stealth virus incorporated RNA of a predecessor of Ochrobacterium, and it makes our conclusions about incorporation of RNA of RNA sequence in a viral genome more reliable. As a result and conclusions, what I can say, the first thing when you find an RNA-like sequence in a viral organism, you should check the assembly. The assembly can have lots of mistakes. It can be assembled incorrectly, and probably one of the ways to check it for correctness is to check the presence of RNA. Mycoplasma and host RNA can make our conclusions and make our sequence not reliable. That's the first step. However, in some cases, it looks that some viruses like limestone bacteriophage, like stealth herpes virus, they do contain RNA-like sequences, and they were incorporated. They were incorporated regularly from their hosts, as well as from other members of ecological mission. Thank you for your attention. Any questions?
Вы ему позвоните, скажите об этом. Все хорошо. Все. Понял. Colleagues, hello. Any questions? Okay. Thank you for your presentation. Unfortunately, no questions. Thank you.
All right. Thank you.
And next speaker. Some problems with my. Okay. Sorry. Sorry. Lukyanova Anna. Anna, hello.
Hello, good morning. Can
Good
you
morning.
see me or hear me?
Okay.
You hear me, right?
Yeah, sure.
Nice. Okay, and also you see my presentation,
Yeah,
right?
sure, I see. Yeah.
Okay. So again, good morning, dear colleagues. I would like to present you our research devoted to analysis of updated Pictobacteriaceae sequences and our proposal to taxonomy revisions, which we make according to this analysis. First of all, I would like to say about the object. Pictobacteriaceae is a bacterial family of phytopathogens infecting actually a wide variety of plants, including important agricultural plants such as potatoes, onions, carrots, and so on. And these bacteria infecting crops cause wilting or rotting or another diseases, which leads to gross damage and economical loss. And that's why this family now is actively studied. And despite the classical microbiological and molecular methods still is very important, but the bioinformatical approach actually makes at least equal contribution to this study, and particularly the analysis of sequencing data. And for this group, in open databases, sequencing data accumulating is extremely fast. For example, if in the middle of the last year we had about 250 genomes in a gene bank, now it's more than 400. And in this study, we downloaded new genomes and analyzed them. And so I'd like to present some observations that we did during this analysis. And first of all, we calculated average nucleotide identity of type strains of each pictobacterial taxons and newly published strains. And what conclusions or observation we can do? First of all, look at this group of unclassified pictobacterium strains. According to the values of average nucleotide identity, all of these strains shows quite high similarity between each other and probably forms the one common group. According to this analysis, the closest relative of this group is Pictobacterium aquaticum. But the value of average nucleotide identity with Pictobacterium aquaticum is lower than 96%. That means that probably this group not only forms a separate clade, but this group is probably might be elevated as a new separate species. Second observation we did is focused on this strain of Bruneria L33HA. This strain demonstrates quite low similarity, first of all, with other Brunerias, and secondly, with all other pictobacterial strains. Therefore, we propose, we hypothesize that it's probably was misclassified and this strain is not Bruneria at all. And to make more precise analysis and to prove this data, we did another one analysis using up-to-date bacterial core gene pipeline. This pipeline construct a phylogenetic tree using 92 concatenated sequences of housekeeping genes. So we can get quite precise phylogenetic tree. And according to this method, we also see that discussed earlier Pictobacterium strains really forms the separate clade, and this clade is monophyletic, and the sister group is Pictobacterium aquaticum. So this Pictobacterium group is probably a new species. Secondly, the Bruneria L33HA shows to be probably an ancestral branch to not only for other Brunerias, but to all Pictobacterium species and representatives that we used in this study. This misclassification was probably occurred during the publication of 16S RNA sequence, because 16S in Pictobacteria are quite conservative and oftenly they don't allow to distinguish correctly the isolates, and after publication of 16S RNA with a mistake, subsequent publications were made without any corrections. So in order to do even more precise analysis, we used another one phylogenetic tool. It's bacterial pangenome analysis pipeline. So as it's said in the name of algorithm, it's pangenome analysis, and it's used 1,071 core protein genes, and actually makes very accurate phylogenetic tree. And as we can see, this tree has the similar topology as the previous one, and probably it is correct. So as a result of this work, we published like an update of pictobacterial taxonomy according to new published genomes. And we propose that a group of pictobacterium strains will be probably evaluated to a separate species. And second point that Brunneria L33HA was probably misclassified, and we propose that this is ancestral line to all other Pictobacteriaceae taxons. So thank you for your attention.
Thank you for your presentation. Somebody has questions? No questions. Thank you again. Next speaker, Takmakova Anna.
Yeah, I'm here. Can you hear me?
Yes, yes, I hear you. Please start your presentation.
Good morning, dear colleagues. My name is Anna Takmakova. I would like to present you our research, first insight on the decay box PCR fingerprints and its suitability for strain clustering. First of all, I want to say what is a decay. Decay is a genus of phytopathogenic bacteria that cause soft rot of plants. Also, it's one of the most negative potato pathogens. In the figure 1, we can see the effects of this pathogen on potato. Because of this pathogen, about 20–25% of world's agriculture harvest is lost annually, which is colossal economic damage for each country. An innovative solution to this problem can be the treatment of seed and wear tubes with bacteriophage suspension. This method of protection against pathogenic bacteria will be ecological and safe for people and the environment. However, due to the narrow specificity of phages and the huge number of species of the genus, we need a diagnostic system that would allow us to quickly determine the type of pathogen. Also, such a system would avoid expensive and labor-intensive sequencing. As such a diagnostic system, we tested box PCR method. For the box PCR method is one of the types of genomic fingerprinting. High resolution make it possible to distinguish bacteria at strain level. The distribution pattern of amplified fragments in the gel is unique for each strain. So we tested this method on 2 species of genus Dicea, on Dicea solani and Dicea dianticola. As we can see, the distribution pattern for both species turned out to be different. The result showed that all 4 strains Dicea solani showed a common band distribution pattern in the electrophoresis. PCR products were distributed in 6 bands ranging in size from approximately 20 base pair to 1720 base pair. The distribution pattern of amplified fragments in the gel had the same appearance for all samples. Comparison of box PCR table values banding patterns showed that all 4 representatives of the species had the following bands: 418, 518, 618, and 1100 base pairs. Also, 3 out of 4 representatives had a band of about 300 base pairs, and 2 out of 4 representatives had a band of 1700 base pairs. To obtain fingerprints of Dicea dianticola, 4 strands were used determined by 16th as rRNA. PCR products were distributed in 4 till 9 bands, ranging in size from approximately 116 base pair to 2100 base pair. The table shows that all 4 strains had a band headbands of about 500 base pair and 900 base pair. Most of the representatives had headbands of about 400 base pair, 1,200 base pair, and 1,400 base pair. For GALE image analysis was used the software application system PAL. Using that software, a dendrogram was built. The neighbor joining method implemented in the PAL software clustered the fingerprints into several clades. As we can see, the clade containing the decayed anticola strains features significant diversity. To check the reliability of this program, of this dendrogram shown on the slide, we carried out phylogenetic analysis. To do this, we sequenced 3 strains from our experiments: decayed Selene F020, decayed anticola F085, and decayed anticola F90, and the rest of the data was taken from the open source. The phylogenetic analysis conducted with 16 sRNA sequences of different type species belonging to the genus of decay and several strains of decayed Selene and decayed anticola places F085 and F019 strains in one clade with decayed anticola strains, but also demonstrated a very diversity within this group. Decayed Selene F020 is clustered with the other decayed Selene strains. So this dendrogram fully confirms the result of our first dendrogram based on the result of box PCR electrophoresis. The box PCR method has long been used to establish phylogenetic relationships between strains and differentiate existing and de novo isolates. After obtaining the box results and checking the effectiveness of this method using dendrogram construction and phylogenetic data, it can be concluded that this method can be used for the clusterization of the decay strains. And also it is simple that the whole genome sequencing. By this method, patterns were obtained that make it possible to clearly distinguish decay Selene from decay anticola. Thanks for attention.
Okay, thank you for your presentation. Any questions? No questions. Okay, what next? We will have a break till 2 o'clock, 2 p.m. in Novosibirsk time. Yeah, just a moment. Yeah, sure. Okay, we will have a break during 45 minutes. It will be a long break. Okay, thank you. I will wait you in after 2 o'clock in Novosibirsk. Okay, dear colleagues, we are starting our work, and next speaker is Evgenia Amelina. Please, could you present your talk?
Yes, hello. Okay. Can you see my presentation?
Yes, sure.
Yes, okay. Dear colleagues, let me tell you about our work with 3D visualization of brain tumors via artificial intelligence. This is the result of 2 years of our research project, which was funded by the Russian Foundation for Basic Research. Our project is dedicated to the use of artificial intelligence for non-invasive diagnostics of neuro-oncological diseases. The background of our project meets some trends. The number of neuro-oncological patients increases constantly. For Novosibirsk region, about 400 patients per year and up to 2,000 or 3,000 patients with brain metastasis. Now, MRI is the gold standard for detecting brain tumors, marking the surgical field and postoperative monitoring in neuro-oncology. As a result, the ROC stress load for medics, radiologists, neurologists, neuro-oncologists also increases. And the inevitable consequence is the risk of so-called human error and medical error. And one of the possible approaches to solve mentioned problems is the development of new diagnostic methods based on artificial intelligence systems. These challenges require the involvement of highly qualified specialists in various fields: radiologists, neurologists, neurosurgeons, oncologists, mathematicians, data scientists, programmers, and so on. It's necessary to create large data sets with specially annotated images. And here I've listed the main types of tasks that artificial intelligence can solve when analyzing MRI images with a suspected cancer process. Tumor detection is the first step. We want to know about the presence or absence of tumor and its localization. The party concerned a neurologist and oncologist. The segmentation task provides the clinicians with information about tumor and edema boundaries and tumor's volumes. The classification task allows us to make an assumption about the type of tumor, its classification, and so on. Additional requirements for the quality of image processing are put forward by modern treatment technologies. For example, the use of a gamma knife requires preliminary marking of the tumor, and the development of